Frolova, and today I'll be talking about my poster titled Applying Rearrangement Distances to Enable Plasmid Epidemiology with Pling. The first third of this is based on already published work, so please do check out the paper and the actual tool. But first of all, what is the problem overall? We're interested in analysing plasmid genomes, but these can be very problematic because plasmids undergo frequent rearrangements and gene gain and loss, and SNP analysis is often a lot less informative than it is in, for example, bacterial genomes. So what you need is a model that actually reflects this and how plasmids evolve through structural change. And so that was our core underlying idea, and the model that we selected is something called the double cut and join. So this is illustrated here in this figure, but basically in this operation you do a double cut and then you can join the ends again in two different ways, and this simple operation will basically describe any rearrangement that you can think of, and then in combination with indels, that's all of the structural changes that a plasmid can undergo. So, having this model, what we did initially is use it to cluster plasmids. So the core idea is that you encode the relatedness of plasmids in a network and then cluster on its basis. In this relatedness network, each node represents a plasmid, and to begin with, you draw an edge between two plasmids if their containment distance is low enough, i.e. if enough of the smaller plasmid is contained in the larger. So this gives us a measure of sequence similarity, and this is sort of our initial filter on the plasmids. And then after that, for all the pairs that are connected on the containment network, we calculate these DCJ indel distances. And then we remove any edge for which these DCJ indel distances are greater than 4, which means at this point, any two plasmids are connected if they have enough sequence similarity and few evolutionary changes between them. Now, the trouble with this is that you can get something called a hub plasmid, or we call them a hub plasmid, and basically these are really densely connected plasmids whose neighbors are not. And usually this happens is because this very densely connected plasmid is dominated by a promiscuous transposon, which is creating this false signal of relatedness. So then we filter that out, and then finally we have this network that really represents the relationships between the plasmids, and we're able to cluster on them. Basically what we found is that this seems to work pretty well. I'm not going to go into the benchmarking, it's onto the paper. So having this, we wanted to go further with the idea of studying structural change. And our overall goal was to compare rates of SNPs to rates of structural change so that we can actually get a sense of how quickly do plasmids change. And so for this, we're trying to focus on populations of plasmids that are sort of already circulating and are being vertically inherited rather than ending up in a population through horizontal transfer. And in order to isolate out these populations, what we did is, of course, cluster with Pling. Then you can map the distribution of the plasmid types identified by Pling on the host phylogeny. And from these distributions, you can find the clades in the host for which the plasmid is highly prevalent, right? So for instance, here, you've got a clade, and then the idea is that, you know, in this clade then, the plasmid is being vertically inherited. So the overall tree structure is the same for both the plasmid and its host. And we know the host tree structure it's given. We've inferred it using SNPs. And so what we can do then is basically prune the tips with the plasmid out of the overall tree. So we know the general structure of it, but we don't know the lengths of the branches. And in order to get that, we can do ancestral reconstruction. And we do this ancestral reconstruction with SNPs, and we do it with these DCJ indel distances. And then, if you divide the branch lengths by time, you get these root tip rates per year of SNPs versus DCJ indel. And here they're plotted for a bunch of different plasmid types as identified with pling. Each color is one type. The size of each dot is relative to the core genome size within that subcommunity. And you can see that the distributions of structural change, aka DCJ indel, versus SNPs is very much dependent on plasmid type. So we see there's some very distinctly structural change heavy subcommunities, and then others that are more prone to SNPs. And we even see that there's some plasmid types that are undergoing really very, very little change and are remarkably conserved. And thank you to my wonderful collaborators, in particular Leonard, who's really the backbone for all the theory on DCJ indel, for helping with this work.